Kia ora everyone, my name is Michael Riley, and over the summer I worked on a project investigating forests and nursery plant pathogens in New Zealand with a focus on Phytophthora and Colletotrichum species. In this project, I focused on an initial dual culture screening, testing different oomycete, bacteria, yeast, and fungi potential biological control agents against Phytophthora and Colletotrichum. The project was supervised by Dr. Rebecca McDougal and formed part of a larger study led by Professor Ian Dickey. The core and the first trial of the study was a initial dual culture assay where I put a pathogen and a biological control agent on agar plates and assessed the antagonism and categorized it as either volatile or physical. Volatile, as you can see on the bottom there, the pathogen is growing away from the BCA and they are avoiding contact. Physical is above the volatile one. The BCA overgrew the pathogen and inhibited its growth through physical contact. So if the trials that were determined to be volatile, they were further assessed using stacked plate assays, where one plate was inoculated with the pathogen and another with the biological control agent. Then they were stacked one above the other, allowing them to exchange volatile compounds, however, not being able to physically touch each other. And the growth of the pathogen along with the biological control agent was measured as they grew out. The other pairs that were determined to be physical were further assessed using water agar slides. In this method, one edge of the water agar slide was inoculated with the pathogen, while the other edge was inoculated with the biological control agent. And then as the isolates grew through the media, their interactions were assessed using the microscope to clearly see is the biological control encapsulating the pathogen or are they just growing alongside each other. Then to compare the biological control agents against traditional methods, there were a series of agar plates prepared with different concentrations of copper and phosphate in them. And then the pathogens were inoculated onto these concentrations. And again, the measurements were grown to compare against the stack plates, the initial dual culture assays, and the control plates. So across all screening methods, antagonistic interactions were highly variable and pathogen specific for most species. However, one bacterial isolate consistently inhibited multiple pathogens, which is my suggestion for Dr. Rebecca to carry on through for further trials. In addition to this laboratory screening, I also gained experience with a variety of laboratory equipment and molecular techniques, along with a range of fieldwork activities. These included DNA extraction and PCR sequencing to learn more about some of the isolates Dr. Rebecca McDougal provided for me, and a seedling assays for Colletotrichum affected pine in a controlled environment. I also had a trip to Pureora where we inspected tōtara trees for Phytophthora podocarpi and set stream baits up to test what Phytophthora species might be in the stream there. We also spent time interacting with stakeholders. All in all, every step that I was exposed to demonstrated to me how applied plant pathology integrates field ecology, laboratory experimentation, and molecular tools. giving me a better perspective of how science is applied in the real world. Finally, I would like to acknowledge and thank Bioprotection Aotearoa for funding this project, along with Rebecca, Corinne, and the rest of the BSI Rotorua team, who were always willing to help when I was stuck and made this project a fun and rewarding learning experience.
Questions for Michael? Ian, Dickie, and then Peter.
Kind of nice talk. I'm just curious about the evolution here. Do you think these bacteria are evolved to inhibit the pathogen, or do you think it just is a random thing that happens? Is there a reason it would select for it as a behavior?
I would assume they are, because with bacteria was mostly we were seeing the volatile effects there, and so that the volatile was essentially stopping the pathogens from affecting the growth of the bacteria. And through the PCR sequencing, they were also associated with other bacteria which have been known to inhibit the pathogen growth. So I would say that the bacteria isn't feeding off the pathogens, but I would assume the volatiles are through evolution because it's given them space to grow themselves and get the food source which they are living on for themselves rather than sharing it with the bacteria.
Cool, thank you.
I was going to ask how many of them were interacting through volatiles, but it sounds like actually that seems to be a big component, that volatiles seem to be having a major effect. Have you got good ways of identifying what those volatiles are that are actually causing that interaction?
No, yeah, we didn't get into that. We were just mostly determining the volatiles and then measuring that effect. Dr. Rebecca McDougal can hopefully pursue and maybe figure that out in further trials.
Brilliant. Thank you.
Thank you.